Our fourth and final speaker is Dr. Suzanne Topalian. She's going to be presenting our early phase trial of the new PD-1 targeted immune therapy.
Today I'd like to describe to you the clinical activity, the safety, and a potential biomarker of clinical response to the drug Anti-PD-1, which is an antibody therapy. PD-1, or programmed death 1, is a molecule that's expressed on the surface of activated immune cells, and it has a very important role in suppressing anti-tumor immunity. In order to understand how anti-PD-1 works, you need to understand a little bit about how the immune system works and how it can fight cancer. T cells are a central cell type in the immune system that fight cancer. T cell function is regulated by two different signals. Signal 1 is a specificity signal whereby the T cell recognizes its target. And here we're talking about the target being components of tumor cells. But then you need a second signal to tell the T cells what to do, a regulatory signal. And that signal can be positive or negative. If the signal is positive or stimulatory, the T cells become activated. They secrete cytokines. They can kill tumor cells. They proliferate. They percolate throughout the body, seeking out and destroying tumor cells. And all of that is what we want to see. But after activation, T cells naturally begin to express the molecule PD-1 on their surface. And this will turn T cells off if they encounter the partner molecule PD-L1 or PD-ligand 1. Tumor cells can express PD-L1. And so the interaction between these two molecules creates a protective shield that shields the tumor from immune attack. So even if the T cells can recognize the tumor, they can get to the tumor. Once they get there, if they're expressing PD-1, if the tumor is expressing PD-L1, the T cells will be turned off. Anti-PD-1 antibody is a blocking antibody against PD-1, and so it interrupts this interaction. It functions to rescue exhausted T cells and to enhance anti-tumor immunity. The phase 1 trial of anti-PD-1 that I'm describing today was a multi-dose regimen in which the drug was given in the outpatient clinic once every 2 weeks. Patients were treated for a cycle of 4 treatments over 8 weeks, at the end of which they were restaged. Patients were eligible for this trial if they had advanced metastatic melanoma, kidney cancer, lung cancer, colorectal cancer, or prostate cancer with progressive disease after having at least one other cancer therapy, but they were allowed to have up to 5 other therapies. In general, the patients who came on this trial had good performance status, but they were heavily pretreated, so approximately half of them had had at least 3 other cancer therapies before they came onto this trial. After the first cycle of treatment, if patients had rapid progressive disease or clinical deterioration, they went off study. If we encountered unacceptable side effects, the patient remained on study. They did not receive any more drug, but they continued under observation. If the patient demonstrated tumor regression, stable disease, or even if they had some progressive disease but they were clinically stable, we continued to treat those patients until we either saw a confirmed complete response, worsening progressive disease, or unacceptable toxicity. And we could treat patients on this trial continuously for 2 years after they went into a follow-up phase. Here I'm showing you the drug-related adverse events or side effects that occurred in at least 5% of 296 patients, which is the total patient population on this trial. You can see that serious side effects were encountered in 14% of patients, and the most common side effects are listed here. There are other side effects that are not listed because they occurred less frequently. Many of these side effects were consistent with an immune-related causality, as you might expect if you release the brakes on immune responses. As we're seeing anti-tumor responses, you might also see immune-related side effects. We did see 3 treatment-related deaths on this study in 1% of the total patient population due to pneumonitis or lung inflammation, which we believe has an immune-related etiology. And over the course of time, we have, I think, developed better ways to identify people who are at risk for this side effect, and also better ways to detect it early on and to treat it aggressively. Also note that only 5% of all the patients treated on this trial have discontinued treatment due to related side effects. So in general, the treatment was well tolerated in an outpatient setting, and in general, the side effects were manageable. This is showing the clinical activity of anti-PD-1 antibody in 3 different types of cancer across a wide range of doses. The largest number of patients in this treatment population, that included 236 patients for which we had at least 6 months of follow-up, were melanoma patients. And in this population, we saw 26% of patients with objective responses. And objective response means either a complete regression or a significant partial regression of cancer. We also saw stable disease that lasted for at least 6 months in another 6% of patients. Among lung cancer patients, and this includes patients with squamous as well as non-squamous subtypes, we saw a CR plus PR rate of 18%, and again, with another group of patients who had prolonged stable disease. And finally, in kidney cancer, 27% response rate and 27% prolonged stable disease. There were 31 patients on this trial who had a response that occurred at least 1 year ago. And among those 31 patients, 2/3 of them had a response that persisted for more than 1 year. So one of the remarkable features about this therapy is that it can induce very durable responses in otherwise treatment-refractory patients with advanced disease. We did not observe any objective responses in 19 colon cancer or 13 prostate cancer patients. And finally, I'd like to draw your attention to a possible molecular marker that would allow us to predict which patients are most likely to respond to therapy. In a subset of 42 patients on this trial, we examined pre-treatment tumor biopsies for the presence of PD-L1, and again, this is the partner molecule to PD-1 that is expressed on tumor cells. And what we found was a correlation between PD-L1 expression on tumor cells, and here I'm showing you the staining of these pre-treatment biopsies, an example of melanoma with the cell surface ringed expression, an example of kidney cancer, an example of lung cancer. When we saw this kind of expression, in that group of patients, we had a 36% objective response rate. If we did not see that expression on the surface of tumor cells, we had no responders. I would stress that these are very preliminary data, but give us an important lead for further investigations and potential biomarker development. So in conclusion, anti-PD-1 antibody, BMS-936558, can be administered safely in an outpatient setting to heavily pretreated patients with durable clinical benefit in patients with lung cancer, melanoma, and kidney cancer. These results will be released tomorrow, as you know, in a paper in the New England Journal of Medicine, which is under embargo until early tomorrow morning. And at the same time, in New England Journal, there's a companion paper with a blocking antibody against PD-L1. The lead author on that paper is here today, Dr. Julie Bramer from Johns Hopkins, and she'll be available to answer questions at the end of the session. We found responses also in melanoma, lung cancer, and kidney cancer with a blocking antibody against PD-L1. So we feel that these two studies are, in a sense, bookends. They both point to the importance of the PD-1 pathway in cancer therapy across multiple histologies. The preliminary data correlating PD-L1 expression in pretreatment tumor biopsies with outcomes needs to be further explored, and that's an area of active investigation. And finally, controlled clinical registration trials of this drug in patients with the 3 types of cancer that seem to respond are planned. Thank you for your attention.